Well, thank you all for coming. It was a great pleasure to introduce to you. You're great. I have some notes. You see, what happened is just pure serendipity, because when I was working in Boston in Harvard Medical, I mean, this guy came to my lab and would say, well, one, I'm still Quay and I'm interested in our medicine. And you could ask what our medicine is. It's polypeptide exchange in bilayers. And he said to me, well, why don't we work together? Because I can put together a medicine with lipids and test an hypothesis. Okay. All this I'm telling you, I mean, we published a couple of papers, but the nice things of collaboration, it was that I was just arriving to the medical school, and I was thinking what I'm going to do for a grant. So here appears Steve Quay, give me the idea. So obviously the idea, I wrote the grant, and I got my first engagement with Steve's idea. So I not much recognizing what is doing success, no? So that was my first presentation. Then Steve Quay disappeared. Okay, and now I realized he went to Stanford, got tenure, but there is this thing of adventure. Okay, and he decided to throw away tenure and invent companies. Okay, so he keep doing all the time. I mean, putting together companies, doing extremely great things. I mean, he has an immense amount of papers, but also an immense amount of patents. But patents that produce things. And well, I mean, when he's surviving, he got a salary for that. And a salary that allows him to come to Chile to see his best friend. And I mean, it's a pleasure to have you here, Stephen. So, I mean, for years.
Thank you very much. Okay.
It's a pleasure to be here.
It's 2 a.m. in the morning, and a 34-year-old Stephen Quay is lying inside a magnet, the largest magnet ever created by man, about 3.5 million times the dipole of the Earth's magnetic field. I'm lying inside this magnet in Long Beach, California. I've traveled down from Stanford by plane in order to be injected, to be the first human to be injected with gadolinium, which is way down at the bottom of the periodic table, very deadly. It's 0.01 angstroms different from calcium in diameter, and of course, 3+, not 2+. So if it finds a calcium channel, it goes in and it sticks and you're dead. So I'm hoping that I made the chemical right in the laboratory. But and the reason I went to Long Beach was this was very early in MRI technology. I mean, we're not very many magnets in the world. And I'm there at 2 a.m. in the morning because the magnets cost $10 US a minute to own. And so during the day, my friend who's a radiologist was doing patients, you know, brain tumors and all sorts of things. So at 2 a.m., the machine was available. So I'm lying inside the machine. You know, I've been lying in these machines for a couple of years, so I can go to sleep pretty easily. But Keith Burnett, my radiologist, calls in, he says, because he's looking at my body without contrast, and then he's going to inject me with gadolinium. So before the injection, he said, Steve, did someone ever tell you there's something wrong with your left kidney? I said, oh, Keith, stop bullshitting. Yeah, sorry. No, there's nothing. What are you doing? Why are you teasing me? He said, no, there's this really big mass in your kidney. Looks like it might be cancer or something. What do you want to do? I said, well, inject me with the gadolinium. If it lights up, I'm going to be dead in a year. And if it doesn't, then we'll see what happens. And I'm obviously here and I'm not 36. So I'll tell you how that ends in a little while. But this is why I'm sometimes called Valley Doc. And this talk is lessons learned from a life in Silicon Valley spent between calcium and gadolinium. Excuse me. Now my daughter in the back is a sophomore in high school, and she's scrunching down now. But anyway, we just went through a year of chemistry. and the periodic table. So this is sort of dedicated to her. I'm going to talk about things I've done in what I call the 3 different kinds of sciences. So there's hypothesis-driven discovery science, which I consider pure science. You make observations and you have a question, you create a hypothesis and then you test it, and you can either verify the hypothesis or reject it. I mean, it's nice and clean. The second kind is what I call observation-based science, glorified bird watching. You get some really cool equipment with new technology and you make observations, but it's not hypothesis-driven. And then finally, there's the inventive or need-driven science, biotechnology, where you identify a need, you find a solution, you imagine a solution, and then you synthesize, formulate, test, and deliver. So when I came here, I realized that the last hypothesis-driven science I did in my life was with Ramon. Because after this, I went into biotechnology and the inventive process. So I want to review what we did together, because in 2 years and 2 papers, we found some interesting things. Hopefully, most of you know about the fluid mosaic model, which is about 10 years old when we were working on it. But the basic idea is that the proteins in the membrane, that this is a semi-liquid state, depending on the temperature and the composition, and the proteins can move up and down based on voltage and other properties. So that was this was the field we wanted to test things in. And as Ramon said, what I had developed with Karana at MIT was a set of phospholipid analogs that had these carbene precursors, either nitrines or carbines, which can actually separate a carbon-hydrogen bond. Because the idea was we wanted to photolyze with light, cause the carbene to be formed, and then it would insert itself wherever it was. So the idea is you have let's say a transmembrane protein, you hit it with laser light, and then you can look at what amino acid was cross-linked, because it can cross-link anything. It doesn't require a charge to do its action. So these were the various classes of lipids, and then we moved it up and down the fatty acid chain. C6, C12, C16. So could you incorporate these photolabels in living cells? Well, you could. So Gobind and I grew these with the fatty acids on a fatty acid oxytrope, an E. coli that required fatty acids for growth. And you can actually see that, you can, I'm probably still radioactive from these because this was sulfur, I think. So I did about 1,000 of these gels. So these are the membrane proteins separated by pH and isoelectric point in the native E. coli membrane. And then when you do photolysis with a C12 fatty acid, you actually see a subset of these proteins. So only a small number of proteins were actually able to be cross-linked in this position. We were able to show that the cross-linking had to occur at least at C6 or lower in the half leaflet of the bilayer. So the question that I then came to Ramon with was, could we distinguish surface versus inserted configurations of this peptide alomethicin, which comes from spiders? So it's designed to cause pain in your neurons when you're injected with it. But it's a very convenient peptide analog for voltage-dependent channels. So at the time, the models were that it laid on the surface or it formed channels or maybe a dipole at an angle like this. But the hypothesis would be that if you photolized if you photolized a fatty acid here with a label and it didn't label the alomethicin, then that would be an indication that it's probably laying on the surface. But if you can actually cross-link it, then it means it's in the membrane. And in fact, so we would label alomethicin so you could find it in a column process. We put the synthetic lipid with the carbene precursor here. We sonicated to form vesicles. Now, in these vesicles, there's no voltage, of course, because it's made from lipid mixtures of alomethicin. But in fact, after cross-linking and then separation, you can actually see. So we have a chemical reaction that chemically broke alomethicin at this position. So you could have full-length alomethicin or this portion, 1 through 14, or 14 through 21. And you can see actually that there is a new peak here in the presence after photolysis that shows that has both phosphate, showing it has phospholipid associated, and radioactivity. So it's radioactive from the L-methicillin. And then with we had FedEx back then. You know, it was a long time ago, but we did have FedEx. So I would FedEx the photo product to Ramon back at Harvard, and he was able to show that you could actually form voltage-dependent channels. They had individual states in the conductance. The conductance was less voltage and less concentration dependent, but otherwise was very similar to natural L-methods. So with these sets of experiments, we were able to reject the surface configuration, which was satisfying. And I think further work has demonstrated that this is correct. Yes. So at this point, then, as Ramon said, I got a little impatient, or whatever the word would be, and went off into biotechnology. And since I have invented 7 drugs that are approved by the FDA, this is the gadolinium that I gave myself. This is an echocardiogram of a colloidal dispersion that forms bubbles in the bloodstream. Here's a plain echo. So this is the mitral valve. This is the ventricle. Looks pretty normal there. But then with the contrast, you see this huge blood clot. So this person is about to have a stroke. And you couldn't see it in the plain film, but you can see it. Now I'll talk about lessons I've learned along the way, because as much as you like to think that you learn from the things that are successful, you usually learn from your mistakes in life. And boy, I've made my few. So lesson 1, problems are not the problem. So this was my first company. I called it Salutar, which is pretty close to your health. And so that's the drug. And these are the various investors I had along the way. Eventually, we had to sell it to Nycomed and General Electric. But at the time here, in the spring of 1983, this man, John Papajohn, who was one of the major financiers of biotechnology, although he lived in Iowa and most of biotech was on the West Coast at that point in time, said, I will invest $600,000 in your company. I'm going to fly my private jet to San Francisco. We'll have a meeting at the airport. I'll give you my check and you're good to go. And I'm obviously pretty excited at this point in time. But what I have 2 days to think about it. And what I think about is, gosh, imagine what I could do with $2 million, you know, and just not 600,000, but 2 million. All the, you know, if 600,000 gets me here, 2 million will get me here. And so I'm a genius. I got this. Let's go. So the meeting starts. I'm telling you what I can do with $2 million. And I suddenly hear from the back of the room, this meeting is over. Son, I can handle problems. I can handle setbacks. I can handle challenges. What I can't handle are surprises. I came here to write a check to you for $600,000 and now you want $2 million. Thank you for your time and good luck. Now, at this time, there was an American folk singer named John Denver who wrote a very nice song called Leaving on a Jet Plane, because that's what John did. Now, I ended up being successful and he did call me later and say he had made a mistake. But nonetheless, the lesson I learned with problems are not the problem, surprises are. So for your graduate students, postdocs, or people that want to go into biotech, keep your stakeholders informed every day. Don't ever surprise them with a setback if you can see it coming. Lesson 2, the magnet and the man. So what happened to that guy in the magnet back then? So I raised this money finally for my company. And the animal experiments are going great. This is a mouse inside a smaller version of the magnet. But I'm running out of money. It's 600,000. Even in those days, it wasn't enough. Running out of time. To go to the next stage, next important point, I need FDA approval, hospital approval, medical school approval, ethics committee approval, board of directors approval, finding patients, etc. And nobody wanted another mouse experiment, especially the mouse. So what to do at that point in time? Well, why don't we give the drug to this guy? So I decided to give the drug to myself. And that's what I did. So I went into the magnet. I remember I was told I had a tumor. And in fact, this is what was going on in my left kidney. Now, there is this thing called a benign cyst. So basically, it's a cul-de-sac in your glomeruli, in your tubules, that forms during fetal development. And it's a cul-de-sac, so you have this pressure your whole life, and the cul-de-sac gets bigger and bigger and bigger. So right now, when I'm talking to you, I have about 1,000 ml cysts here. I can't get a doctor to drain it, and it's benign. It doesn't hurt me in any way. What it was useful for was this. I raised $7.5 million with that picture. So I'm pretty happy with that. And, you know, if I hadn't had that cyst, who knows? Now, 6 months later, the FDA called. They had heard about it. They were not happy that I had experimented on myself. So I sometimes pretend to be a lawyer. So I said, well, look, I made the drug in California. I tested it in California. You're a federal agency. This is a state. And they said, well, that's cool. But where was the magnet made? Oh, Wisconsin. So the federal government, anything that crosses a state line, they get, anyway. So I said, never do it again. I said, I'm sorry, yes, sir, never again. You're the boss, etc. So lesson 2, sometimes it's better to ask forgiveness than permission. Okay, lesson 3, the rule of 43, because no is never no. So this is Stephen Harrison. He helped launch Chicken Soup for the Soul, which is a very popular self-help book kind of thing. Rich Dad, Poor Dad is probably the #1 finance book in the self-help areas. So Stephen Harrison's answer was that silence is never no when you're trying to sell something or promote something to yourself. So when he and I were giving some lectures pre-COVID. This was actually, October pre-COVID, just before, in New York City. And so, so I do what I call my head answer. So I like statistics a lot. So it's a way to test things in the world. And so when you do a coin flip, how many times do you have to get a heads in a row until you know the coin is rigged? Or in business, how many times do you have to pitch your new business until you can conclude it's a bad idea? So if you think about each coin toss being a 50-50, when you do 7 coin tosses, it's a 1% chance of getting heads, all heads in a row. So you need 7 heads in a row to know that 99% certainty that the coin is rigged or that your idea is a bad idea. So when you're pitching a business, you know, if you send 7 emails or you talk to 7 VCs and you get no, excuse me, there's a 99% chance they don't want to hear from you and your business idea is a bad one. The question is, are you the one who continues trying, because there's also a 1% chance they do want to hear from you. In fact, so what happened was that when I decided to develop chemicals, dyes for MRI, the venture capitalists, I went to every venture capitalist up and down in the Stanford area, and they all said, this machine makes such amazing pictures, no one will need a dye. And I kept saying, because this machine makes such amazing pictures, a dye will let you make amazing movies inside the body, physiology. So I went to 42 venture capitalists who said no. Okay, so what's the chance of getting 42 heads in a row? Well, it's roughly 1 in 1,000 billion. But I was persistent or crazy or something. Venture capitalist number 43 said yes, and it turned out to be Arthur Rock. Now, you may not know his name. He invented this word, venture capital. He invented it. He's still alive. He's about 87 or 88. He was the VC for Steve Jobs, the same year that I was involved with him. He financed Intel, a little chip company, Apple Computer, of course, and Genentech. And so 80 million people around the world have used this dye to make movies inside their body. Arthur made 2,700 return on his investment in the company. The epilogue, okay, this is funny. So years later, I asked Arthur why he invested in me. Was it the science you liked or because we both studied at Harvard? The reason I invested in you, Steve, was because when I asked you how many VCs had told you no, you said 42. I went to only 35 people for my first venture investors. That told me you might be crazy, but you were definitely determined. As for going to Harvard, that was a negative. You're the first guy I've met from there with a fire in his belly. So how many times do you follow up? Steve Harrison says silence is never no. 7 is the number your head says with a 1% chance of persistence, but my heart said 43. The rule of 43 is because no is never no until you quit.
Just always to remember. Now, when I developed those photo-labeled fatty acids, I was working with Govind Khurana, who got the '68 Nobel Prize for the genetic code. Govind's rule, which is very important, is that big problems and little problems take the same amount of work. So you might as well work on big problems. So here's an example of what he did in thinking about that. So he developed all of the chemistry for nucleic acids. So if you think of PCR or SARS-CoV-2, anything involving connections between two nucleotides, he did it. Now in those days, he didn't patent any of it, which it's an interesting idea of what was going on there. But in any case, so he had two choices. He could make a simple piece of DNA and show that it works in a test tube, which would have proved that he could make living things, or he could make a complex piece of DNA and show it works in a cell. Okay, so it took over 200 people years of work to make this tRNA, which is what he was making, and it attached an amino acid in a cell that didn't have the tRNA. So that was his test. It took 50 people 4 years. Now, this is the girl in the back there a little while ago. It now can be done over a weekend at Taipei American School, where she goes in Taiwan, by my 15-year-old working alone. So the machines have now been developed to do the same thing over a weekend. But that's how science develops. But because he picked the problem of the humble genius who built the first synthetic genes, he saw that if he could do it in a living cell, people would say he's created life. And that's a controversial concept, but nonetheless, it's a big concept. Doing it in a test tube would have been another test tube experiment. And this is actually after he got the Nobel Prize for the genetic code. So lesson number 5, the first industrial revolution was Henry Ford and the assembly line. I grew up in Michigan, very close to this, so we were motor car people, so to speak. And the idea is that the assembly line allows one person to do the same thing over and over again, but it greatly accelerated product development and those kinds of things. Now, the second revolution to me was an idea that Stephen Jobs had. We used to have to go once a month. We had to go up to San Francisco to have dinner with Arthur Rock. Tell him what we were doing. This is no fax machines, those sorts of things. And the guy had a memory. So if you said you're going to do something 3 months ago, he would remember. So we would drive up there together. And so Steve said, here's the crazy ones, the misfits, the rebels, the troublemakers, the round pegs in the square holes, the ones who see things differently. They're not fond of rules. You can quote them, disagree with them, glorify or vilify them, but the only thing, you can't do is ignore them, because they change things. They push the human race forward. So while some may see them as crazy, we see genius, because the ones who are crazy enough to think that they change the world are the ones who do. He had a benign cancer. He wasn't, I talked to him at the time. I said, he needed to have it removed, and he didn't. He tried diets and things, and eventually he died of a benign cancer, didn't have to die. But what he did was he created this Kool-Aid, that the idea that a bunch of kids in sneakers can change the world. Cetus, Microsoft, Apple, all of these companies, Genentech down here, all these companies were founded by scientists just like you, thinking that they can change the world. Of course, they did. So at my first company, Salutar, the venture capitalists decided I was too hyperactive to deal with. I didn't run. Now, maybe they said I was crazy. I'm not sure. They might have said I was crazy. So my employment agreement actually said to get my bonus at the end of the year, I needed to run 60 kilometers a week. Because if I ran that much, I was kind of mellow. But otherwise, I was too hyperactive. So I now have knee problems because of it. But anyway, this is the 68 New York Marathon. I did beat the lady in the garbage bag because I heard her coming at the finish line.
You're wearing pretty much shape there, huh?
Are you saying I'm not now? Are you saying? Yes, that was back there. I'm bleeding from the, I'm bleeding here. That was painful. And this is my last marathon in Taipei.
This is interesting because it's a back and forth. So there's a bus. Because Taipei is a big, big city. So there's a bus that's going at a 4-hour and 15-minute pace. And if the bus gets to you, you have to get on the bus. So I'm running here and the bus is going there and I'm going, because I'm not that good. So rule 5 is either be crazy or stay home. Okay. Rule 6, venture capitalists and pies. So a little bit of understanding of how they work, how venture capitalists think. What is the smallest amount of money to create a change in value? And then how much equity, how much of the company do I need to control for this investment? So with my first company, I wanted 8 million to get FDA approval. 600,000 got me a mouse picture. But I wanted to sell 15% of the company. They wanted 51% because they always want control. So how a venture capitalist sees a piece of pie is they see a half of the pie. They want to have control of the investment. And so, in fact, Arthur Rock patented that method. He went to 40 companies to invest in Intel. They all said no. So he bought 51% of Intel for $2.5 million. It became 126 billion in 2019. So what do you do? So in my next company, I asked for 3 million to get an IND because I already had mouse pictures. I still gave up 51% of the company, but I was able to then take it public with just that amount of money, raising money in Europe and Japan, and then doing a public offering. First one in biotech from a Series A investment. So lesson 6 is 100% of 0 is 0. So you do whatever you have to do. You give whatever percent you need to get your money to get your company started. Because if you just sit here at the starting line, you'll sit there at the starting line. The last lesson, lesson 7. This is Bob Mumford. Who I had a meeting with founder of Crosspoint Ventures in Sand Hill Road, which is where all the venture capital sat. Remember, this is before cell phones and all sorts of things. So I show up at his office, and he says, "Oh, I'm sorry, I couldn't reach you. Bob is at the hospital with his wife. They're having their first baby. Would you like to reschedule?" I said, "No, I'll go to the hospital. I'll talk to you, at the hospital." So I head to the hospital. I stop in the gift shop. And I buy a bib and a little outfit for a child. And I sit there and I pitch him in the waiting room. And he's running back and forth. This is before the time when dad stayed in the delivery room the whole time. Bad idea, because women forget the pain and all that, and the men are just traumatized for years. But separate story. But anyway, so he's going back and forth, and I'm pitching him, and I'm thinking, the day your child is born is like one of the most wonderful days of your life. It really is. So I think he's gonna invest it. I'm part of this and all that sorts of things. Well, no. He said no. So fast forward to my next company. It's 1990, and I'm starting my second one. I get a call from Bob. He said, "Hi, Steve. Bob Mumford here. I heard you're starting another company. Why don't you stop by and tell us what you're up to?" So I go to the meeting. There's like 12 people in the meeting, and here's Bob. Now I had to, this is Photoshop, full disclosure, but he's wearing an 8-year-old bib. And it really is 8 years old because it's gray, it's got yellow spots, spit up and everything. And he's wearing the bib. And they leave the investment. So a $10 bib becomes a $3 million investment, and Bob made $69 million on that bib. So this last lesson, where's the last lesson? Is it here? Oh yeah, the last lesson. It's always personal. Just keep that in mind. So those are the 3 kinds of science. I have returned lately to hypothesis-driven discovery science in my spare time, I guess, because I've been testifying both at the State Department and the U.S. Congress twice on the origin of SARS-CoV-2. And without any, going to the lab or anything, you can look inside the genome. And you can see that this was an engineered virus. It was an accidental lab leak, but nonetheless, the virus was created in a laboratory. Probably the Wuhan Institute of Virology is my bet. So thank you very much for your time.
I have many questions, but the first one is about how you went from the kidney issue to having all that money. You went very quick.
Well, yes, this is entertainment. This is not a science section. So, I was down there and we saw the lesion. We saw this large lesion on my left side here. And so if it doesn't light up, it means that the blood flow, the glomerulus is not filling this. So these 1 out of 3 people has a cyst in your kidney in this room. So it's very common and benign, never causes any cancer. So, so when it didn't light up, I knew it was a benign cyst, but nonetheless, I could sell it to the venture capitalists. Here's what you can see, because you can see blood flow in the normal kidney around it, but not the cyst. So you can tell if it lit up, then it would be a cancerous cyst, and I would have had to lose the kidney.
So the other question had to do with the process by which you create an idea. Do you have, how does that happen? Do you observe the world or you purposefully?
Yeah, so
ideas? How does that happen?
the question, how do you come up with ideas? I mean, to me, the most important thing is to have as broad a base of science understanding as you can, because the inventions and everything are in the overlap of maybe 2 different fields. I had this crazy accident when I was a graduate student in Michigan, where if I went to the library to read a journal article, I copied the article before and the article afterwards, and I read them. And they, of course, were, they're not, in journals, they're not in crazy order. But within a couple months, other graduate students would say, I got this problem, and I would remember something I had read. And so my whole life until the internet, I would take an inch of articles home on weekends. I'd sit on my patio, I'd have a glass of wine, I'd read articles, and it's just a matter of many different things. The other thing is the gadolinium product that I invented would not have been invented by a chelation chemist, because he or she would have said it was impossible, it wouldn't work. I did some chemistry on it that the traditional chelation people said wouldn't work, and it did work for a very strange reason. And then the bubble program in the ultrasound program was the same thing. Because no one said you can make a fluorocarbon emulsion that was stable. Oswald's ripening is the physics process from 100 years ago that said I couldn't make it. But it turned out by adding a very high molecular weight, high soluble oil, I could violate Oswald's law. So the other thing I found was that if you're too deep in a field, the assumptions in your field hinder you in being able to see new things. So it's kind of reading all kinds of crazy stuff and then going into a new field.
You have to be on focus. You have to be on focus.
On focus, yes, on focus. But it's also inefficient, so you need to be very dedicated. My wife will tell you too dedicated.
More questions?
Yes.
It went along to me that you didn't mention anything about contacts, because
About what?
contacts, having contacts, because how did you end up with all these people that you approach, these 42 people?
Oh,
You didn't receive any recommendation for.
Yeah, so, Silicon Valley is the home of venture capital. And I think everyone knows this term, Silicon Valley, and they've tried to replicate it around the world with some very successful different places. But being at Stanford, there was just an ecosystem, even in 1980. So, for example, I was at a swimming club on the weekend, just going swimming, and the fellow next to me was Larry Sonsini. So Wilson Sonsini is the law firm that started every biotech company in the world, and we're just chatting, over lunch on Saturday. So he starts introducing them to venture capitalists. When you come out of Stanford and go towards the freeway west, there's the SLAC linear accelerator on the left, and on the right is something called Sand Hill Road. And 90% of the venture capital in the world is there. It's it's a very small spot. It's maybe about 3 acres. You just don't go to the and you have to take no. I got a lot of no's. I mean, I always wonder because I almost quit. I almost quit trying. I couldn't,
I'm curious about your last comment.
well,
it's like the conclusion on what we read in the news is that it's not very clear whether it's man-made or it's
Yeah, yeah. So I'm happy to talk about that. I don't like to get into politics too much. Now, unfortunately, it was politicized, right? But a zoonosis is when an animal in nature has an infection of a microbe or a virus, and it infects a human. So every zoonosis has an animal and a human and a virus. In natural spillovers, you can find the animal pretty quickly. So SARS-1 took 4 months to find the civet cats in the markets. MERS took 11 months to find the camels. And now after 3 years, we've tested 80,000 animals, 400 species. So no intermediate host has been found. So that's one of them. Point number two is, so I'm sorry, you look at each compartment, animal, human, virus, and test whether it came from nature or life. So in a natural spillover, the virus is optimized for an animal and practices jumping into humans and then going back until it gets the mutations to really go in humans. So these practice jumps cause asymptomatic infections, but you can find antibodies later on. So with SARS-1 or with MERS, they would go to the blood bank and take blood out from like years before the epidemic and find antibodies. And so about 1 to 2% of the general population had SARS-1. 20% of the people working in the wet markets had antibodies to SARS-1. So they did 36,000 blood bank specimens in Wuhan, where this began, and zero seroconversions. So this virus was not practicing on humans at all. And then the virus itself has what's called posterior diversity. So think of the virus as every 2 weeks, when it's somewhere in a population, it's adding a new mutation, 2 a month. So with SARS-1 or with MERS, when you looked at the first 100 jumps into humans, there was a lot of diversity. And then you could build one of those trees and you could walk it back. So you could see that SARS-1 went into the civet cats about 18 months before the human epidemic began, because there's diversity. With MERS, it was 5 years. And with SARS-2, every virus that any of us have had came from one person. There's no posterior diversity for SARS-2. So the furin cleavage site is very unusual. It's never been seen in 1,000 years in the subgenera cervical viruses. So 1,000 years, it's never happened. And then the genetic code for it, so it's an RRAR, that's the amino acids, but the code is CGG. That has never happened in cervical viruses ever. That's the arginine codon. CGG is the arginine codon. And coronaviruses hate using the CGG. There are 6 different codons for arginine, and they use one of the other ones in very high proportion. But in the laboratory, when you're doing cloning, you use CGG. So there's that. The virus was optimized for humans. So there's 200 amino acids in the receptor binding domain. So there's 20 possibilities. So 4,000 possible positions in the receptor binding domain. And a guy named Jesse Bloom at the University of Washington changed every one of those to see which ones improved binding, which ones didn't improve binding. Only 17 out of 4,000 made it a better binder. So it was 99% perfected for humans when it first started. And then there's something called ORF8, which does 2 very bad things. It suppresses interferon response, so you're asymptomatic, which is, there's never been a new respiratory virus in humans that is asymptomatic. Because it's hitting a new immune system, it's going crazy, the immune system is going off like fireworks. But ORF8 suppresses the interferon response. It also suppresses MHC presentation, so you can't make antibodies to it. So this whole problem of getting reinfected or the problem of factor vaccines don't work very well is a product of this thing called ORF8. And it has 2 engineering sites on the end of it. So when you do synthetic biology, you move things around with restriction sites. And there's 2 restriction sites on either side of it.
The sequence are not found in other viruses?
No. ORF8 is a completely unique protein. Now it has completely, SARS-1 has a protein that's 10% homologous. MERS doesn't have one at all, 0. But you can see in PhD and master's thesis, so what you guys are doing right now, you can see at the Wuhan Institute of Virology, they were playing with ORF8 for about 5 years. Unfortunately, that's one of the gain of function attributes that is bioweapon. So you're not allowed, when you gain a function, you can change infectivity, transmissibility, or pathogenicity. You can't change asymptomatic transmission, make it hard to detect, or antibody formation, make it hard to get antibodies against. HIV is the poster child for suppressing what's called MHC presentation of antigens. So it's why you can't make a response to HIV. It's like your immune system on marijuana. It's just so.
This is very strong disease.
Yes, yeah, I'm writing a book. So I was consulting with the State Department in 2020, and then with the House of Representatives in 2021, and the Senate last year. And I'll be helping with testimony this spring. In the House of Representatives.
So, okay, virus has no so many origin.
There's no what?
So then it's not so.
Do not, yes. Well, so just to be clear, a synthetic virus like SARS-CoV-2 is 95% from nature, and it's from bats. It's only the 5% that they take. So it's all, I mean, it's not it's 30,000 letters long. Maybe the nucleic acid is wrong. You don't start with a piece of paper and write out 30,000 letters and you make it. We're not that smart.
It's not fully synthetic.
It's not fully synthetic. It's 5% of that. That's all you need.
Okay, but in a way, if you have a partially,
Okay, what?
partially synthetic, in this case,
Yes.
5%,
Correct.
can you think about it as a bio?
Yes.
Yes.
First of all, thank you for your presentation. Very good. Second of all, sorry that the last slide has taken the spotlight because I'm also going to ask about COVID. But,
I didn't want to.
do you think that, I want your opinion, assuming that it is a man-made leak.
Accidental man-made leak is what I mean.
Do you think that it would make life more likely because it shows how possible it is, or less likely because it shows how difficult to control it is? Because the most effective country.
That's a non-scientific question, and it's a really good.
That's why I asked for your opinion.
It's a really good. Well, so I'm writing a book on this, and in the last section, I'm making recommendations of new laws for gain-of-function research, because I think one of the other things that I found was that in December 2019, one astrologer was working on Nipah virus with synthetic clones. So they're cloning the Nipah virus, which is against the Biological Weapons Treaty because it's 80% lethal. So Nipah and Ebola are just, they're racing to be number one, number two in killing humans. And here in 2019, the laboratory is working on Nipah virus. So I'm hoping that new laws around gain-of-function research can be added to protect us. Because this virus is 0.5% lethal overall, but a 10% virus, a 15% virus, 200 years of a setback. So maybe back to 1800s.
I have a question, general thing about your first part of all this great environment that you have in Silicon Valley.
Yes.
That allows you to have
Yes.
this, create these companies with science and all that we don't have. I mean, we don't have in Chile. So what do you think? How it was created, that environment? It was kind of like it was planned to be like this. It was like something like was serendipity in the sense, or because American people are so willing to take risks to do business and they give money to do.
Yes. So just to be fair, the Silicon Valley in California they tried to replicate it in Boston and in St. Louis and in San Diego and in Germany. It hasn't happened. So it's not a matter of Chile or anything. It just it got started. And it got started there when we went from vacuum tubes to transistors. That was the transition. And there were 6 people at a vacuum company that wanted to work on transistors in the vacuum company. They wouldn't let them. And they met Arthur Rock, and so they started National Semiconductor, and then they transitioned to Intel. But that really started it. And then once you have, you need lawyers, and then you need accountants. And so very quickly, but well, 20 years, within 20 years, when I arrived in 1980, they had been 15 years in the business of starting companies with lawyers, and with accountants, and all of the work out now, you know, it was done then. So it just happened there. Now it's continued there, and it's now 100 times bigger than it was when I was there. I mean, there's crazy amounts of money, and they're losing money sometimes.
But I guess probably the whole American system, if you think the history, I mean, Tesla went to US.
Yes.
They with the Westinghouse company and everything. So I think it's in the history, I mean, the beginning of United States allowed
Perhaps.
something that we have to think about this because
Well, yes, and I think the biggest lesson, biggest difference in Silicon Valley, at least in Europe, is if you fail at something and they understand your failure, that's a positive for you because you've learned from your failures. I know at least in Germany, when I talk to German scientists and things, you know, you can't possibly fail. Failing is failing. And so it is a mentality where they like you to fail. You fail fast, you fail quickly, you move on. I have 7 FDA-approved drugs, but I have 25 drugs I've tried. So I have 18 that didn't work.
because here we don't have this environment.
Yeah.
So here, if we want to do something more like applying science,
Yeah.
it's the government that sort of control that. I mean,
Yeah.
the government create the environment, they give you small amount of money for do some research, associated with some company. Most of these studies never go to the market because they
Yeah.
fail in some parts. And so I think we're kind of like in a wheel and it's not really going to anywhere.
Yeah, I mean, maybe look at the industries that are really established here, like maybe the wine industry, or maybe these, and the palm oil, or, you know, and talk to those people and say, hey, what's the medical applications maybe of some of this, and they can help support it. I mean, that's kind of what happened in Silicon Valley. I mean, they took a little idea and then ran it. So you start with what you have, or maybe your minerals, your mineral resources.
My question? I just like to comment. I feel very close to your beginning with the gadolinium.
Okay,
Because I was injected gadolinium to detect the tumor in my blood.
that's my good.
Okay, and also because gadolinium blocks any channels, which is the field that I work.
Yes.
Okay, so it was very interesting to know that from something that you call blood calcium channels, but many of the channels are parallel to calcium, so it's not just calcium channel.
Correct.
Okay.
Yeah.
Calcium channel are the ones that are parallel just to calcium. There are many other channels that are parallel to which are less selective,
Yes,
parallel to molecules. And now we found here these non-selective channels are very important in many chronic diseases. And it's like magic. We found by scientific means
Yes.
that the molecule that's generated by the endemic tree in Chile blocks this channel.
Okay.
And not just one, it blocks 3 or 4. And but we cannot get money to
And does it get into the bloodstream?
yes.
You see,
It goes into the brain everywhere. So,
you take it orally and or IV.
either way.
Okay, wow.
But then it's just what Agustin was saying. We don't have the environment to find entrepreneurs and industry that would be interested in developing.
That's
So,
fine.
because we are not, we don't have a way to keep paying for the patent that
Yes.
is approved. Okay,
Yes,
so
and the risk.
it's a waste of money.
Yes.
For us, okay.
Yeah. You should talk. I mean,
You're in the mouse stage.
Well, actually, the human, we're testing the human
Did you really?
yeah. And people that is non-treatable disease, they get better. So even people that are wearing wheelchair, now they're walking.
okay,
Okay,
interesting.
I have a picture that they sent me.
Okay.
And
Maybe we can connect. Thank you.
you. But
Well, Stevens, thanks a lot. I mean, there's several lessons for the students. I mean, the first one, that you can put forward an immense amount of information and interesting information in half an hour. Okay? So you don't need to give those long talks to your speaker. Okay? The third thing that I think is extremely important for you, despite what my dear director and so on.
Be careful when you say that.
I think you can do it. And, if you're crazy enough and you take a big program, I mean, that's the question. Okay. So, I mean, I really appreciate it because you transmit so many interesting and you interpret me so well. I mean, in this, you need to be absolutely bananas and crazy to do what you do.
Yes,
But the other,
brothers by a different mother.
but the other important thing, you can see Stephen's career. I mean, he's a physician and then went to learn chemistry, essentially, you know. But between chemistry and physics, I mean, it's a mixture of all that, that if you want to do something interesting in applied science, I mean, you need to have all those, I mean, expertises. So that's another lesson. Okay?
Physician,
And
scientist.
the spy that, you know, I stopped in the first part of your talk, right? I mean, I'm doing basic science, okay? But it's so nice when you see somebody telling you that, I mean, applied science, I mean, can be really interesting because it's a nexus between, I mean, knowledge and what the people receive in health and things like that. So thank you so much, Jackie. Thank you. It's a small present for you. This is part of our life and our dreams, because it's the story of the building, and maybe when I am dead, it's going to be okay. Okay. It's the story of what the building is going to be. Okay. Thank you. Thank you very much. So.